So hello. It's a very it's my pleasure to host this talk in the seminar series on applications of geometry and topology, the GEOTOP-A seminar. So thank you to the organizers for inviting me to host today's speaker, who is Lida Kanari from the University of Oxford, who will talk about from neurons to networks and understanding the brain through algebraic topology. So thank you, Lida.
Thank you very much for the introduction, and thank you for giving me the opportunity to talk about my work to a more broad audience. So I will start by explaining the neuroscientific aspects of this work, and then I will go into the mathematics, the algebraic topology. But feel free to ask questions if something is not clear. So understanding the brain from the perspective of neuroscience requires a lot of different scales. So computationally, there are all the different scales ranging from molecular DNA all the way to brain structures and whole brain regions. And one of the fundamental questions is, because we don't know how to combine these computational scales, we don't know which parts of the brain are important for giving rise to the cognitive properties that we see at the higher level. So is it all the way through the molecules that we need to understand each single molecular contribution, or can we go to different levels and understand the networks independently of smaller structures? And today I will focus more on somewhere in the middle. So neuronal morphologies are these very intricate shapes in the brain, which is the tree structures of the neurons. And in neuroscience, it's a very well-known person, Santiago Ramón y Cajal, who has been drawing these pictures of brain regions and individual neurons by hand. And he was the first one to observe these very complicated structures and start hypothesizing about which parts of these structures are important for brain functionality. And of course, we have progressed quite a bit since these times of hand-drawn neurons. So now we have digital reconstructions of these neuronal morphologies. And now we can get a better picture of all the different types of neuronal shapes that we encounter in the brain. So here are a few examples of different branching structures within the rodent brain. And as you can see, there is quite big variety of these neuronal shapes. So very much like the botanical trees that we can observe in nature, and we have all these branching patterns, we see something similar in the brain. And this is just a small snapshot of the whole variability that we observe in biology, because in different species, we have even larger differences between the shapes of neurons. And I'm going to show you today a little bit more about that. But one of the fundamental questions with respect to neuronal morphologies is if this variance that we observe of all the different branching shapes is significant for the functionality of the brain. Or is it just a byproduct of random processes that result based on how the neurons are generated in the brain during development? So let's start by a very simple task. Let's try to differentiate these two structures. And as a mathematician, when the neuroscientists gave me these two types of cells, and they said, these are fundamentally different, but how can we separate them mathematically, that was quite a challenge for me because I could not really see immediately what is different in these structures. And what neuroscientists typically do is they measure different properties within these tree structures. So they start counting the lengths of the branches, the number of the branches, the angles between them. But if you do that, the problem is that you have a choice of the features that you select. And therefore, you can concentrate on different types of information. So you lose the full picture of the neurons. So when I started studying these types of objects, I wanted to create a descriptor that would preserve as many properties of these trees as possible. So the solution came from topological data analysis. And here is a quite general example of what we typically mean by TDA, which is focusing on different aspects of the data, so on different scales. And in order to do that, the common input for these type of algorithms is a point cloud. So we have points in space and we let circles around them. And then as we start growing these circles, we start seeing interactions between them. So every time we have intersections between these circles, we add points and we generate the simplicial complexes between these points. So this kind of traverses the point cloud through different sizes of lenses in which we can start understanding what are the interactions between the points. And as we start having intersections between these circles, and there are a little bit different types of algorithms of how to construct them, but we start observing higher level structures. So we have triangles and pyramids and even higher level structures. But this is a quite general view of point clouds. I wanted to create something specific for trees. So how can we transfer this idea of decomposing a point cloud into the fundamental topological components into a tree shape? So we'll start with something very, very simple, which is just this tree structure. And we will assign the leaves and the bifurcation points in this tree. What is very important is that we have a well-defined root in this type of neuronal trees. So the root is given from the experimental data in which we have with quite good accuracy. So since we have this root, we can define any type of function on top of this tree structure. And here, because we have straight lines, you can imagine that we could have some type of function like the radial distance from the soma. So every point gets assigned a property which corresponds here in this particular example in the radial distance from the soma. And what we're going to do is to compare the radial distances of the different leaves from the soma. So as we start taking each one of these leaves and following them backwards, we're going to compare the values of these functions, so in this case, the radial distance between two leaves that bifurcate at the same point. So they merge together here. And we see that the highest radial distance is defined by this point B. So what we do is we follow this point all the way through the root and we generate one bar from the first time we saw this component all the way to the root. Then we go to the next point and we decompose it until the bifurcation point that it merged to the largest component. And so on and so forth. So iteratively, we go through all the leaves and we generate for each one of these parts of the tree, different bars. So this is called the persistence barcode. Now, once we have the persistence barcode, we can take advantage of all the TDA tools that have been developed to study these type of objects. So the easiest type of representation that we can study is the persistence diagram. So we take the start and the end points of these bars and we put them in a two-dimensional space. So we transform these points until we have an equivalent representation. So this is the persistence diagram, and we keep track of the first time and the last time we saw each component in the underlying structure. And now the advantage of this representation is that it can be easily transformed into a vectorized version. So this is one example of a vectorization that I'm going to use today, which is called persistence image, introduced by Adams et al. In 2018. And here we just put Gaussian kernels around the points, and we generate some type of density field based on how many points we have in the persistence diagram. So this gives us a very nice representation of the original tree. So here is an example of the whole process. So we start from the dendritic tree, we decompose it into this persistence barcode, from which we can generate diagrams and then persistent images as Gaussian kernels. Here is an actual biological example of a neuron within the mouse brain, and we keep a color code of the longest components to the smallest one. So we see we go from deepest red all the way to deep blue, going from the branches that have the highest radial distances to the branches that have the smallest. And it's pretty nice to see in the persistence diagram all these different properties transformed from the original tree. And then we can get the persistent image, which is a vectorized version of the same type of descriptor. So how can we understand a little bit more intuitively the properties of these type of diagrams? So you can imagine that at 0, we have the root of the tree. First of all, the number of points that we have in this persistence diagram correspond to the number of branches in the tree. So we can retrieve the number of leaves in the original tree. Then the distance from the central point is how close we are to the root of the tree. And what biologists call the oblique part of the tree is all the branches that are close to the root. And then as we go far away from the root, we have what the biologists call the apical tufts. So we see a decomposition of the branches between the proximal and the distal parts of these neuronal trees. And then another important property is the distance from the diagonal. So the points that are far away from the diagonal are the ones that persist the longest, because in this case, we have a small start radial distance and a big end radial distance. So these are the branches that are extending far away in the tree. And then every point that is very close to the diagonal, those are the small branches that have a short lifetime. So typically, in persistent homology, the components we want to keep are the ones that are far from the diagonal, because those are the more persistent. But you're going to see quite quickly that this is not the case in neuronal trees, because what in point clouds is considered as noise, here we have significant information. So a large number of small components corresponds to branches in the tree that are biologically significant. So let's go back to our original example. We wanted to compare these two trees. Now let's see if we use this transformation that I described, what will happen. And here are the persistence images of the two trees. I hope now it's much more obvious what's the difference between the trees, and you don't need to be a biologist to understand what is fundamentally different. So what we immediately see from these persistent images is that there is the existence of a secondary cluster in these type of trees, which the biologists call the tuft of the apical dendrite. So these are called dendrites, and they are the parts of the biological neurons that are receiving information. They are receiving electrical signal from other neurons. And this second part, this cluster, is quite obvious now if we compare the persistence images. But the advantage of the topological description of the trees is that now if we subtract one image from the other, we can precisely identify what are the differences. So obviously we see the difference in the second cluster, but we see a secondary effect which was not obvious just by looking at the trees. So we see that the branches in this cell type are also extending further compared to the branches in this one, for the branches that correspond to the ones that are close to the soma. And you're going to see later on how much an effect this has in biological structures. So that's a good start. We have a very nice, robust mathematical descriptor. What can we do with it? And here are just some examples of how we used this descriptor in the past. So use it for classifying cells. And these are different cell types in the rat cortex. And we managed to distinguish them quite nicely with a very high classification accuracy, just based on their persistence images. But another type of application is clustering. And this is quite significant because in many cases, the classes of the neurons are not known. So this is an example of an amazing data set from the human brain. So for the first time in 2017, we started having data from the human brain. And because these data were quite novel, there was no class assignment by the experts. And we took these neuronal trees, we extracted the persistent images, and we saw that there is a significant difference between two groups in these type of neurons. So what we saw was pretty similar to what I already described. So some of the cells had the existence of a second cluster of branches. And these type of cells were bifurcating quite earlier in the tree structure, and they were having these very large and extended trees. And that was very different from these other type of trees, which were having just these small branches far away from the root. So there were two different classes of cells that we were able to distinguish just based on their topological shapes. But this is quite important for the neuroscientific aspect because for this type of data set, we also had information about their functional roles. So what we discovered in this paper is that there is a significant difference in the functional roles between these two classes that we've identified based on their shapes. So these type of neurons, which are called profuse neurons, fire at higher rates. So they have an electrical activity that is quite distinct from this other type of neurons, which are called slim tufted cells. And that was quite important because we still don't have a biological answer of why exactly we see this difference. We just know that the topology of the neuronal shapes has a significant impact on their functionality. Another line of application is how we can use these type of descriptors for other cell types. And a great data set that we got from a collaborator in IST Austria from Sandra Sigurd's group was some microglia. So together with Catherine, we've analyzed these microglia from the lens of the topological data analysis, and we found out that there is a significant difference between the persistent images of different microglia from different brain regions. And this data set was quite important because, first of all, we had a very large biological sample. So we had 20,000 cells from all these different regions. But we also had a distinction between the male and the female shapes of the microglia. So one of the aspects that we discovered is that the male and the female microglia in the brain have very different shapes, and we can separate them. And even more importantly, because this data set corresponded to healthy as well as disease cells, so they knocked out some genes in some of the mice that created some pathological condition, and then they observed how the microglia were recovering in the brain as the animal was developing. So what we've seen is that the male and the female mice were having very different trajectories in how they were recovering from this pathological condition. So this obviously has significant implications for the biology because it tells us that there is a different process taking place in the two different sexes. So what can TDA tell us about being human? Most of the data I presented today were coming from rodent, from mice, or from rat. But there has been this notion in neuroscience that the human brain is quite special. So what is the fundamental difference between the human brain and the other brains that makes us perform cognitively as we are? And this question is becoming even more significant recently that we have some artificial neural networks, LLMs, that can perform as good or even better than humans in some specific tasks, even though they have very different architectures. So can we identify what is fundamentally different in the network structures of the human brain? And can we take advantage of that to create artificial networks that have similar properties? In order to understand this problem better, we need to start by the data that we have. And we have a lot of biological data at different scales. So we have reconstructions of the human and the mouse. We have information about the density of cells in the two species. And we also have information quite recently about the connectomic properties. So how neurons connect in the two species. So what we did is put experimental data together and create computational models for the two species. So we know experimentally that the human cortex is thicker, so it's quite larger. There is a significant difference between the neuron density in the mouse and in the human. So in the human, we have a smaller number of cells within the same volume. So we have a lower cell density. And then there is also a significant difference in the distribution of cell types. So most of the cells I discussed today were pyramidal cells with these very complicated apical dendrites. But we also have a different type of cells, which are interneurons. And the main functional difference between these cell types is that interneurons inhibit activity, so they lower the activity of the neurons they connect to, and pyramidal cells are excitatory, so they excite the neurons they connect to. So there are very fundamentally different dynamical properties between these two cell types, but they also have significantly different topological shapes. And the percentage of interneurons in the human brain increases quite a lot. So this is something that we certainly need to take into account in a computational model. So now we can compare also the scales. So we see obviously the human cells are much larger. And then when we put all this information together, we can see that overall, within the same volume, we have the same length of neurons. So this is important because we have a smaller number of cells, but they are in the human brain, but they are larger. So overall, they are covering the same space as the mouse. So if we build the networks between these two species, we can start to analyze them computationally. And here we build a similar size of a human and a mouse network. And then we can do any type of analysis on these networks. Here is a random selection of 100 neurons, and we see already that there is a significant difference in the networks that they form. But in order to analyze them consistently, we take advantage of another algebraic topology property, which is the definition of simplices. So normally, when we have one simplice, we have two neurons connected, and this is the undirected case of simplices. But now, in this particular case, we want to introduce directionality. So in the undirected case, if we had one simplex, we would have a combination of two neurons, two simplex, three neurons, and so on and so forth, but we didn't care about the directionality of the connections. Here, what we need is in each one of these structures to have exactly one source and one target. So one of the neurons that is participating will be the source and one would be the target. So this means that the information are transmitted towards the same direction. So any neuron that is participating in this second degree simplex is transmitting information from the source to the target. So this works like a highway transmitting information towards one direction. And this is quite interesting because they've seen initially that in the blue brain models, we had a significant increase of the number and degree of simplices compared to random networks. But then this analysis was generalized to data from other species. So in microns, which is from mouse cells, we observe exactly the same structure. So we have a higher complexity in the networks compared to any type of random networks, which is the same in other species. So there seems to be something significantly important about this structure that makes the biological networks generate way more of these structures compared to any random network. So we're going to take advantage of this type of description to study what is different in the networks of the human and the mouse that we generated. And if we compare the networks between the two species that we created, first of all, we need to reduce the connectivity because when we build this type of computational models, we define the connections based on the touches of one neuron to the other. And this is a quite overestimation of the actual number of connections. So we randomly reduce the number of connections until we reach a biologically relevant connectivity density. And this is based on the connectivity density in the connectivity matrices. So once we have a similar density of connections, we can start comparing their properties. And we see that the pyramidal cells have a similar degree distribution, but the interneurons, these smaller, simpler cells that I described, have a much bigger difference. And this is also coming from the fact that we have way more interneurons. We have 3 times more interneurons in the human context. So how does this reflect to the number of simplices, or to the distribution of simplices? And what we see is that we have a huge increase in the number and order of simplices in the human brain, both in the pyramidal cells and the interneurons. And this is particularly interesting if you observe that these are logarithmic scales, because if we had the actual distributions, the mouse seems to be quite minimal. It seems almost random compared to the human, which is crazy because we have the same connectivity density in the two species. So something is very interesting in the human brain that is creating these higher dimensional structures. And the working hypothesis for neuroscience is there is some type of scaling law, which is very simple. So there is either number of neurons, the size of the brain or the brain region, or even the size of the neurons themselves, that could explain this very complex behavior. But when we try to analyze the differences between the human and the mouse brains by using these topological descriptors that I described before, we observed that the size of these neurons is not sufficient to explain any difference between the two species. So first of all, if we just scale down the human neurons and we make them the same as the mouse neurons, and we just embed them in the same space, what we would expect to see, if the significant difference was just the size between them, would be that the clusters would merge. But we saw that this is not the case, so there are significant differences in the topology of these neurons, that they separate quite nicely. We saw the same in the morphometrics, so even when we scale down the neurons, they didn't have the same distribution of basic properties like length or number of branches. And also the thickness of these neurons was scaling different between the two species. Another property that was very interesting to see that was not aligning between the scaled versions was the pairwise connectivity. So if we take a pair of neurons and we put them at different distances, what is the probability that they will form one connection between their branches? And of course, human cells are much larger, so we were expecting to see larger connection probability between human cells compared to the mouse. But what was quite interesting is when we scale the mouse cells so that they are the same size as the human ones, we were expecting to see an increase into this pairwise connectivity probability. But what we saw was actually the opposite effect. So we saw that there was a drop in the probability to connect between these scaled cells. So those are the largest mouse neurons that are scaled up to the human size, and still they were forming less connections. And our explanation for that is that when you are just uniformly scaling the shapes of these trees, you are also scaling the gaps between the branches, because you're not adding more branches, so the gaps or the holes that are formed within the branches are larger. So now there is a higher probability that anything would come through these without coming close enough to the branches of the neuron to form a connection. So the simple scaling is not sufficient to explain any of these higher dimensional structures that we see in the human brain. So if it's not the scaling law, then what could it be? Let's start by what we've been discussing in this talk, so the shapes of the neurons. So we see a lot of different examples from the human cells and the mouse cells, and then we compare their average persistence images between the two species. And what we see, if we zoom in close enough to the root of these trees, is that there is a significant increase in the number of branches we have in the human cells. So we see that within the same distance, we have way more branches, and also they extend much further in the human case compared to the mouse. So if we look back at the biological properties of the networks that we use, this can be quite easily explained because we have a lower density of neurons in the human brain. So what we have is the cells being further apart. Because we have a lower density, there is a higher distance between the cells. So this creates empty space between the cells in the human cortex, which gives them the opportunity to create more branches within this space close to their centers. So what we see is that just by putting them further apart, we give to these cells the opportunity to create more branches during development. And this seems to have a significant impact at the networks that they form. So let's try to see if this hypothesis is true. What we're going to do is create a hybrid model between the human and the mouse. So we take all the geometric properties of the human cortex, so the size, the length, the distribution of the positions, and the number of cells are preserved, and they are exactly the same as in the human case. But now, instead of putting together human cells, we scale the mouse cells so that they have exactly the same lengths as the human ones. So we create versions of the mouse cells that are as big as the human ones. And once we put them together, what we have is a preserved total length of branches within the same volume. So we have exactly the same properties as geometric properties as the human cortex, but now we have mouse cells covering this space. And what we expect to see is if the geometry of the human cortex was sufficient to explain the differences between the species, we would get an increase in the number of simplices. But if the shapes of the neurons are important, what we would see is the opposite effect. So we would not be able to recover these highly complex networks that we see in the human brain. And interestingly, we see both. So in the case of the pyramidal cells, which is the top row here, we see that we cannot retrieve at all the complexity in the human cortex that we observe. So the orange distribution of simplices, which is the hybrid model, is falling exactly as the mouse one. And this can be explained by the higher complexity of the dendritic branches that I described before. Because if we take topological complexity, which is pretty much just the entropy of these barcodes that we were describing before, we see a huge increase in the human ones, which leads into these complex networks that we've observed. On the contrary, the interneurons that we have, so the simpler shapes, have very similar topological complexity. But when we add more of them, we can retrieve the complexity of the human cortex. So in this case, it is sufficient to have more of these simpler cells to generate higher network complexity. But in the case of the pyramidal cells, we need to have more complex shapes in the individual neurons. And if you compare the degree of these simplices, it's important to note that the pyramidal cells go much higher compared to the interneurons. So here is our hypothesis of how this system works. It seems that in the human cortex, we have this structures of highly interconnected simplices of pyramidal cells, so these excitatory cells. But in order to control this high activity that we have in these excitatory cells, we need more of the interneurons. So we have smaller structures of these interneurons that are controlling the activity of the lower density of pyramidal cells. So overall, these tell us that through the different scales of the brain structure, going from single cells to networks, we need information about the topology of the individual neurons in order to be able to retrieve the properties at the network level. So it's important to study the system as a whole, as opposed to just looking at the network level. So TDA creates a robust descriptor for neuronal morphologies, and we call this descriptor TMD, Topological Morphology Descriptor. This is an important descriptor to be used for classification and clustering tasks, and it also helps us understand the differences between species. So using TDA, we were able to understand what is fundamentally different between the two species. And we've discovered that the topology of the single neurons plays a very important role at the networks that are formed in the human brain. So this gives us a good understanding of how we can use topology to study the different aspects of neuroscientific problems. But if we go a little bit deeper into the topological properties of trees, what can we learn about the specific space of trees? So what can topology tell us about the space of trees itself from a more theoretical perspective? And in order to understand that, we need to go to the inverse TDA problem. So typically that's a very hard problem to solve, because going from the barcodes all the way back to the original data set is not possible when we have an arbitrary data set, such as a point cloud, because there are a lot of different point clouds that could generate the same barcodes. In the case of the trees, it makes the problem a little bit easier. So because the process of going from the trees to the barcodes is quite consistent, we can take advantage of this process and create the inverse algorithm. So here is an example of how we can use the topological descriptor to actually generate a tree that has the same properties, topological properties, as the original tree. You can think of this process as an iterative growth model. So we start from the root of the tree, and at each point we add one small piece of tree. At each addition, we have the choice to continue, so to continue the growth as it is, one branch, to create two branches, so in this case to bifurcate, or to stop the growth altogether, and this would mean terminate the process. And we use the persistence barcode to define the probability to do these three. So originally, when we have one branch, it's just continuing to grow. When we see one new bar, the probability to bifurcate to generate two branches increases exponentially until we reach this branch. And then at this point, we have generated two branches from the original one. So we bifurcated once. Every time we see a new branch, both of the branches that are growing have a probability to create another one. And then every time we see the end of one of these bars, we increase the probability to stop the growth. So this way we assign the continuation, bifurcation, and termination probabilities based on the shapes of the barcodes. So if we go back to this example, we had the biological reconstruction. So this is an actual biological neuron. From this, we get the topological barcode, and then we can generate trees that have exactly the same properties as the original one, topological properties, plus some other information that we keep through the process. But then we generalize to generating a group of neurons that have the same biological properties. And what is interesting here is that by adding some small noise on how the precise branches are generated, we can create unique shapes. So each one of these trees is individually. It has some noise compared to the original tree. We never retrieve the exact original tree, but each one of the generated synthesized cells has the same topological properties. And we can generalize that, so we can generate shapes of all the different cell types that we have. And this is how we can create, for example, a whole cortical column of synthesized dendrites. So each one of these trees is unique into this column. So this is interesting because now we have like the whole circular pipeline. So this is joint work with Adeli and Catherine, and we studied the symmetric properties of this process. So we go from the original barcodes to generated trees, and then we can create the barcodes from these generated trees. And through this process, the topological synthesis and the TMD, we preserve some of the biological properties of the trees. But as I mentioned, we never generate the exact same tree. So one of the questions was, how many different types of trees can we generate based on one barcode? And in order to understand that, we had to create this symmetric group in the representation of the barcodes. So what we do is we order the bars according to their birth time, and then we observe different death times for these bars. Because what is fundamentally important is that from the shape of these barcodes, we can generate very specific trees. And this is coming from the properties of the topological morphology descriptor, because we said that at each bifurcation, we keep the part of the tree that is longer. So once we go to the inverse process, the bar that is generated next within the tree structure can only be glued to anything that is longer than it. So in this particular example, for example, B1, D1 can be glued to B0 because it is fully contained within this bar. On the contrary, B2 cannot be glued to B1 because its death time is coming afterwards. So according to the elder rule that we use, which branch is larger, it cannot connect a larger branch to a smaller branch because then this bar would have been longer. And so on, so forth. So we can see just from the bars in which one bar is contained, where it can be glued into the tree structure. So this way, from the ordering of the death times, we can construct all the trees that are possible to generate based on this barcode. And here we are not accounting for the precise lengths. We are just accounting for this ordering. So this is why we have the equivalence to the symmetric group, because we are just ordering the death times of these barcodes. And we see that in one case where we have this structure, this kind of scale, we can only connect branches to the longest component, so we only get one type of trees. But when we have this triangular structure, we can generate all the different combinations. So in this case, we have very different numbers of trees that can be realized. And we name the number of these trees tree realization number. So each bar has a specific tree realization number, how many trees we can generate with different combinatorial properties from this barcode. And once we count this number of trees, what is interesting is to compare the biological neurons that we get to random barcodes. And what we see is that the tree realization numbers of actual neurons, so the blue are real neurons and the yellow are just random barcodes, is significantly lower for the actual biological trees. So this is telling us that the space of trees that can be accessed by these biological neurons is a subset of all the possible barcodes and all the possible trees that can be generated. And this is one of the open questions because we don't know exactly why and what is so special about these trees. And what is so striking about this property is even if we include all the different cell types and all the different species that we can have, we see that there is a quite consistent structure. So we see significantly lower number of trees that can be realized in the biological actual shapes compared to any random barcode. And another interesting property is coming from the statistics of these barcodes that are coming from neuronal trees. So one very nice study recent by Bobrovsky and Skraba described a universality in TDA. So if we take any type of point process and we generate the distribution based on the persistence diagrams that are coming from these type of point processes, so similar to what I described in the Vietoris-Rips use case, we see that all the different diagrams obey a universal probability law. So when we increase the number of points, they converge to a very specific distribution, which is the Gumbel distribution. And they have shown that this is true for many different types of processes. So from random point processes to actual dynamical systems, even images or sound waves. And they see that all these different types of data converge to the exact same distribution. So what we were expecting to see is that the same would be true for neurons. So can we generate the Gumbel distribution from the persistence diagrams coming from neuronal trees? But this is joint work with Christina Cavazzari, who's now in KTH. And what we found is that neuronal trees seem to be special even in this aspect. So when we generated these type of distributions and we created what we expected to be a Gumbel distribution, we see that there is a significant difference between the neuronal trees. So the cumulative distribution of these diagrams do not converge into the Gumbel distribution. And what is particularly interesting here is that actually they don't converge to any specific distribution. So depending on which cell type these neurons came from, they converge to different distributions. So there doesn't seem to be a universal law that governs the same properties of the persistence diagrams as we get from point processes. And we also generalize this result to DLA. So DLA are diffusion limited aggregates, which is an algorithm that is just generating trees from diffusion. So at each point, you diffuse particles randomly and you generate these type of tree structures. And because this is a completely random process, we were expecting to see something similar to the point clouds. But what we saw is that even in these type of structures, they are more similar to the neurons. So they don't converge to the same universal laws that are followed by point processes. So there seems to be something very specific in the embedding of the actual biological and random trees that is breaking this universality law. So with that, I would like to conclude with some open questions. So if we look at neuronal trees mathematically, we are wondering what is fundamentally unique in these shapes and how we can characterize this tree space that is coming from biological data in terms of symmetry, in terms of universality, and in terms of all the statistical properties that are quite different from any other random process that we observe. And this hopefully will lead to a better understanding between the relationship of the space of trees and the space of barcodes, which of course can be used to answer some neuroscientific questions, such as creating a universal map of neurons, so an embedding that gives us more information about the differences between all the cell types in the brain. And then we can start to link these type of properties to genetic and network information to identify when the system breaks and when we have pathological conditions arising from this process. So with that, I would like to acknowledge the very big team in the Blue Brain project that took part in the computational experiments. Of course, Catherine Hess, who has been collaborating with me in most of these projects, linking algebraic topology with neuronal morphologies. And a special thanks to Adelie and Christina, who worked with me on the more specific projects. And thank you so much for your attention.
Thank you very much, Lida, for this really interesting talk. Yeah, it was very, very interesting. So, well, are there any questions for the for Lida from the audience? I actually have a couple of questions. So the first one is more like abstract, but how sensitive are these results to the choice of metric that you take to compare the diagrams? Because there's no, I mean, there's a choice, right? For example, when you represent the barcodes or the diagrams in these like sort of images. So if you, I mean, so are they very sensitive if you change the metrics that you take?
You mean the metrics to generate the diagrams
No,
or
no,
the
to
metrics
compare.
on the
So
diagrams after
to
the
compare the
comparison? Yeah.
diagrams, right? Because you also have like Wasserstein and other more intrinsic. So
Yes.
bottleneck
Yeah.
or
Yeah. So they are indeed quite sensitive, and it also depends on the specific problem. So we try to see which types of vectorization are better to distinguish cell types. And one example was persistence images. So they were pretty good at picking up the significant differences. With one fundamental difference, so these are unweighted. So normally when you create these images, you weight based on the distance from the diagonal, but because we wanted to preserve the smaller structures, we did not use these weights. And then we saw that there are also some other types of vectorizations that are performing quite good in the distinction. So one that I already mentioned is this entropy. So
Mm-hmm.
the entropy curve is also quite good, or the life curve. When you go to distances, like Wasserstein or bottleneck distance, because they're keeping, like they're weighting more the longer components, we are losing
Mm-hmm.
information and we are no longer able to compare consistently the diagrams.
Okay, so this is a choice you make based on the data,
Mm.
like look
Mm-hmm,
what
mm-hmm,
features you care about. Okay. Yeah, and
mm.
thanks for the answer. Yeah, that's a very yeah, enlightening answer. And with the, I mean, how easy is it to sort of run this? I mean, computationally to run this analysis? Is it like very costly, or does it?
No, it's actually very easy. So we can process lots of data in minutes.
Mm-hmm,
And we actually have the computational code open on GitHub.
mm-hmm,
So we have collaborated with other teams that use the code. It's quite easy to run and computationally efficient,
mm-hmm.
which is good.
Yeah, actually, I mean, maybe going back to the question, when you described, the with the mouse and the hybrid system, and then the human sort of brain, but is there some sort of I mean, if I understood correctly, it's a matter of scale. Like if you rescale things, then they look similar. Is that the idea, or?
So no, it depends on the specific process. So if you just do uniform scaling between the two species, it doesn't work.
Mhm.
Because we have in the human brain, these pyramidal cells have a much larger density of branches
The
close
density.
to the soma. So it's about how the branches are distributed,
Mm.
because
Okay.
if you've seen in the comparison figure in the pyramidal cells, we had all these branches close to the
Mhm.
soma, and the mechanism for that is that anything that passes through this very dense ball of human pyramidal cells has a much higher probability to connect. So
Mm.
that's how we are forming these higher level structures.
Mhm.
Mhm. So
I
in
see.
this case, it's not scaling that is sufficient to work. In the case of the simpler cells, the interneurons that don't have these complex structures, uh, just putting more of them into the same space increases the complexity of the network.
Mhm. And going back to the brain part, I mean, this is sort of, I think you focused a lot on what's different, but can you, I mean, but what is, I mean, the parts that are the same, because the brain is not like homogeneous, right? I think there are certain parts that
Mm-hmm,
change.
mm-hmm, mm-hmm,
And I think the older parts are shared by, say, more animals. And I mean, because
mm.
somehow when you do, is this somehow detected also, or?
So that is quite an interesting and difficult question, because in the human brain, we only have data from a very small region,
Okay.
because they are coming from pathological tissue. So
Mm,
in order to remove that during some surgery, the data that we have are very, very specific.
mm-hmm,
Now, what is interesting is that we are starting to have more data from more species.
mm-hmm,
So actually, yesterday or the day before, there was a new release from monkey cells, which we
mm-hmm,
didn't have so far. So it would be interesting to see how we can try to respond to this question with intermediate species, because obviously we have lots of data from other species. So can we take advantage of that and make a possible prediction for the human brain, because we don't have enough data for the human brain?
mm-hmm. Yeah, very, very interesting. And also maybe, like for example, healthy tissue compared to like
Yeah.
sort
Mm.
of maybe something should also change, I imagine. Yeah.
Mm. Mhm.
Yeah.
Yeah.
Great. Well, thank you. Yeah, this is really fascinating. So, well, I'm not sure whether I don't think the question was clarified on the chat, but yeah, but maybe are there other questions? Well, it looks like it's not the case. So, well, so yeah, there's like time invariant topology preserving mappings.
Okay, so I guess you may be referring to dynamical properties,
I guess
which,
maybe, yeah.
yeah, which is something we have not studied at all for these data sets. This is something we want to see in the future.
As in, like if the brain is, if you grow, right, if you age, also the shape, the connectivity changes, I guess, as you
Yes, so the
are
connectivity
growing and.
certainly changes, and it's actually, we know from other species that the networks are becoming denser with development, and then they start getting sparser as they have specialization in learning. But we have not studied specifically the topology, how the topology changes in terms of time. That would certainly be very interesting to see.
Yeah. There's more. There's another question in the chat. So maybe I will read it. So it says, yeah, thank you for your interesting talk. I wanted to ask about the metrics used in the TMD process. Do you think that any of them would be suitable in providing a reliable metric to compare the similarity between neurons? This is coming from the perspective of studying the diversity between and within neuronal neighborhoods.
So if you are referring to the metrics used to compute the TMD, yes, that's actually quite important. The function that we use, we have not seen a significant difference between the radial or the path distance. So these are the two types of metrics that we typically use and seem to be the best to distinguish neuronal types. If we took other types of information, such as number of branches or some other type of functions, they were not as good in describing the differences. So the choice of the metric is indeed making a big difference in these comparisons.
Thank you. Yeah. So, well, so I guess, yeah, it seems there are no further questions. So, well, thank you very much, Lida, again for this great talk. And thank you everyone for joining. And yeah, I hope to see, we meet again in the next talk of the GEOTOP-A seminar. So thank you.
Thank you very much.